Hello everyone, so we are going to start the part 3. So till part 2 you have come to know how the fatty acids they get synthesized in our human body. So the last product that we have got till now, it's the palmitic acid. So now we are going to start the part 3. So here we are going to deal with some of the basic concept which are actually needed to get understand before the completion of this whole process of the fatty acid synthesis. So we are going to deal with those basic concepts. So let's start. First of all, what is the significance of this FAS? We have already come to know what is FAS, how many enzymes are associated with FAS. Now we are going to deal with the significance, how it is signified in our body. First of all, you should know that this FAS, this enzyme complex, the fatty acid synthase, is actually offers a great efficiency that is free from interference with other cellular reaction. Means all the cellular reaction which whatever it is taking place in your body, they will take place in their own way, but this process it is not under influence of any other processes, so it takes place with a great efficiency. Moreover, there is a good coordination between all the enzymes which are involved in the FAS complex. So there are 7 enzymes about which we have already discussed. So these enzymes, they will have good coordination among themselves. Moreover, now what are the consequences or parameters which are going to regulate the fatty acid synthesis in the human body? So what are those parameters? First of all, you should know that the fatty acid production, it is controlled by enzymes, metabolites and products of the fatty acid synthesis process, sometimes the hormone, even the dietary manipulation. All this, I repeat, the enzyme, the metabolites of the fatty acid, the end products, hormones and dietary manipulation, all this combinedly they regulate the synthesis of the fatty acid. Now first of all, let's start with the enzyme. For enzyme, it is the acetyl coenzyme A carboxylase enzyme. I hope you remember what I have taught you in the first part. So now this enzyme, you see, this is the enzyme, right? It was actually responsible for the conversion of acetyl coenzyme A to malonyl coenzyme A, right? Now you see here the basic concept. So basically this is the active carboxylase, the enzyme, this is the active carboxylase, acetyl coenzyme A carboxylase, it is the active part. Now when this enzyme, it is subjected for AMP activated protein kinase with the addition of ATP, a phosphate molecule will be introduced by the ATP. This ATP which will get converted into ADP, a phosphate molecule will be introduced by this ATP into this active carboxylase, which in turn make it inactivate. You can see here inactivate carboxylase, addition of 1 phosphate. Again, when this product, or sorry, when this enzyme it need to get activated, or when this enzyme it need to get upregulated for the synthesis, then what actually happens? Again, this protein phosphatase 2A, it will remove this inorganic phosphate which have been placed here. You can see here it will remove this inorganic phosphate and finally make it or convert it into the active form. So basically you see these enzymes controls a committed step in the fatty acid synthesis, first of all, right? Moreover, this acetyl coenzyme A carboxylase enzyme, it actually acts as an inactive protomer. That is a monomer, right? It's generally remain as an inactive protomer or an active polymer. So this is the inactive form. Moreover, this citrate, I hope you remember what I have taught you in the first class. So the citrate, it promotes the polymer formation and increase the fatty acid synthesis, right? For further reference, you can again check my first part of the presentation on de novo synthesis of fatty acid. Moreover, the palmitoyl coenzyme A I hope you remember this palmitoyl coenzyme A and the malonyl coenzyme A. These are the two important enzymes. These enzymes, it actually comes in the last step of the fatty acid synthesis, and this is the malonyl coenzyme A. They cause the depolarization. I repeat, it forms the depolarization of the enzyme, and thus it inhibits the fatty acid synthesis. Now, hormonal influences. So what are the hormones which are going to influence the formation of fatty acid? First of all, you need to know one thing, that the hormone actually, they regulate this hormone of your body. They regulate the acetyl coenzyme A carboxylase by a separate mechanism. This mechanism actually, it involves the phosphorylation and dephosphorylation. So the hormone, it regulates the phosphorylation and dephosphorylation of acetyl coenzyme A. So when it undergoes phosphorylation, addition of one phosphate molecule, which I have already shown you in the previous slide, it get converted into the inactive form. And when it undergo dephosphorylation, means removal of the phosphate group, it gets converted into its active form. The enzyme, the acetyl carboxylase or coenzyme A carboxylase, this enzyme, it get converted into its active form. Moreover, other hormones like glucagon, epinephrine, norepinephrine, they actually inactivate. These type of hormones, they actually inactivate the enzyme by CMP dependent phosphorylation by addition of one phosphate molecule. On the other hand, the insulin, it is responsible for dephosphorylation, and thus this insulin, it dephosphorylates and activate the enzyme acetyl coenzyme A carboxylase in your body. Moreover, this insulin actually, it promotes the fatty acid synthesis, while you remember, insulin, it always promotes the fatty acid synthesis, while the glucagon, it inhibits the fatty acid synthesis in the human body. Now, this insulin, it stimulates the uptake of the glucose. We all know that insulin is responsible for lowering the amount of the glucose in the blood, right? Moreover, insulin also promotes the conversion of this pyruvate to acetyl coenzyme A. Thus, this facilitates the fatty acid formation in the human body. Now, what are the dietary regulations? What types of diet are going to influence the fatty acid synthesis process in your body, and what are the other types, or what are the different types of diets which are going to stop the formation of the fatty acid synthesis in your body? First of all, you should know that the consumption of high carbohydrate diet, if you are taking, or if you are on a high carbohydrate diet like your rice, potato, etc., or fat-free diet, the diet which don't have much amount of the fat, it actually increase carbohydrate-free diet, or carbohydrate high diet or fat-free diet, actually increase the synthesis of acetyl coenzyme A carboxylase. You remember this. Moreover, fatty acid synthesis. So these two are responsible for the formation of the fatty acid, which actually promote the fatty acid formation in your body. Moreover, conditions like fasting, if you are on fast, or if you are on a high fat diet, then it decreases the fatty acid production by reducing the synthesis of acetyl coenzyme A carboxylase and fatty acid synthase enzyme multi-enzyme complex. Now, regarding the availability of NADPH, we know what is the role of NADPH, how NADPH it is going to supplement the formation of fatty acid synthesis, about which I have already discussed in my part 2. So we know that the reducing equivalent for fatty acid synthesis are provided by the NADPH, right? Now, which either this NADPH, it either come from the citrate or acetyl coenzyme A transport or HMP pathway. So regarding HMP, you can see my presentation on beta oxidation of fatty acid. Now you should remember that about 50 to 60% of the required NADPH reducing equivalent NADPH, it is obtained always from the HMP shunt in the human body, and which significantly actually influences the fatty acid synthesis. Now elongation and desaturation, how the fatty acid chain it is going to elongate, how it is going to grow in the number of the carbon with the subsequent addition. What is going to add it? Actually the malonyl coenzyme A, it is going to add it, right? So what actually happened, you see here, it's the carbon 16 is subsequently get increases to carbon 18, then to carbon 20. So the consequent addition of the carbon atom to the fatty acid chain. Now this elongation actually it occurs with the enzyme on the cytosolic phase. Elongation it occurs with the enzyme on the cytosolic phase, means basically on the part where of the endoplasmic reticulum membrane, ER membrane. The ER will have two parts, one it is facing inside, another one or another face it is facing towards the cytosolic part, as because the ER it is present in the cytoplasm, right? So the part of the ER which is facing the cytoplasm, on that part elongations occurs. On that part the elongation of this chain it occurs. Now this is done, this elongation it is done by an enzyme, it is known as the elongase, and this elongases actually what they use in every addition. You can see here C16 to C18, right? In every addition actually malonyl coenzyme A it is added, and this malonyl coenzyme A every time it adds 2 carbon subunits. You can see here C16 to C18, then to C20, and subsequently to more number of the carbon. Now synthesis of long chain fatty acid from palmitate. So palmitate, we already come to know how palmitate is formed. I have already discussed in the part 2, right? Now from here new fatty acids are going to form with a new number or more number of carbon. So what actually happens, you see the chain elongations. I already told you it takes place in the cytoplasm, right? So this chain elongation can take place in 2 sites. Actually there are 2 sites in the cytoplasm. One it is the mitochondria, another one it is the endoplasmic reticulum. So actually in the process which actually takes place in the endoplasmic reticulum, those are known as the microsomal process, and the other one it is the mitochondrial part. So these are the 2 processes by which actually it takes place, the elongation of fatty acid chain it takes place. Now you see here the microsomal means the endoplasmic reticulum. Here actually the chain elongation is more predominant. Here the chain elongation is more predominant, which actually signifies more, as because most of the fatty acid they get elongated via this microsomal addition, right? So which involves successive addition of malonyl coenzyme A with the participation of NADPH, which gives you the reducing equivalent. Thus most of the process it takes place in the endoplasmic reticulum itself, or you can say it's the microsomal addition only, right? Now a specific group of enzyme which actually helps in the fatty acid elongation, which I already told you, is the elongases. This elongases enzyme they actually takes place in the fatty acid chain elongation, right? Now the mitochondrial chain elongation is almost the reversal of beta oxidation of fatty acid. I repeat, the mitochondrial elongation. Here I have already told you one more part is there. The elongation can also take place in the mitochondria. It is almost the reversal of beta oxidation of fatty acid. In the beta oxidation of fatty acid, what actually happens? The fatty acid we start breaking it, right? Or here in the mitochondria, right? Or here in the elongation actually it takes place, or it keeps on going, or it keeps on increasing the number of carbon in the fatty acid chain. It also takes place in the mitochondria, and actually this process it is the reversal. It is a reversal or reverse process of the beta oxidation of fatty acid. Now acetyl coenzyme A molecules are successively added to the fatty acid chain to lengthen the chain. We come to know that this acetyl coenzyme A, it is going to keep on adding to the fatty acid chain to increase the number of the carbon in the fatty acid chain, and the reducing equivalent it is always supplied. The reducing equivalent for synthesis of the fatty acid it is always supplied by the NADPH. Now you can see here how actually it takes place in the eukaryotic cell. So this is a overall diagram. You can see here how it keeps on going, or it increases its number. You can see here it's the linoleal coenzyme A. It's a 18 carbon compound. Again it get turned into the gamma linolenoyl coenzyme A. Again elongase will come here. It is going to increase the number of the carbon. It get converted into icosahexaenoyl coenzyme A, which finally get converted into arachidonyl coenzyme A. So in this way, in the eukaryotic cell, the number of carbon in the fatty acids is keep on growing. It's keep on increasing its number in the fatty acids, in the fatty acid chain. So in the eukaryotes, once this palmitic acid it is formed, afterwards more number of carbon it is keep on increasing in the fatty acid chains, just to maintain the demand of the fatty acids in the body. Now you can see here I have shown you a diagram. So here in the previous steps, I am again showing you in the previous step. Actually here it's the arachidonyl coenzyme A, right? This is the end products. Its number is having 20, it is having 20 number of carbon, right? Now from this is the arachidonate. This arachidonate can be also from the diacyl glycerol. This arachidonate is generally also from the diacyl glycerol. So the enzyme which is responsible for formation of arachidonate from the diacyl glycerol, it is nothing but actually it is the diacyl glycerol lipase enzyme, right? And from the phospholipids also this arachidonate is forms in the body, and from the phospholipids the enzyme actually the phospholipase A2, PLA2. This is the enzyme which is responsible for conversion of this phospholipid to arachidonate. Now this arachidonate actually it is the precursor, I repeat, arachidonate it is the precursor for synthesis of most of the cell signaling molecule. I repeat, arachidonate it is the precursor for synthesis of most of the cell signaling molecule in the human body. See, from here, from the arachidonate, with the help of the enzyme lipoxygenase, you can get the leukotrienes, right? This is the cell signaling molecule. From here, with the help of the enzyme, from arachidonate, with the help of the enzyme prostaglandin synthase, you can get the prostaglandin. These are some of the cell signaling messenger, or you can say cell signaling molecules in the body. From the prostaglandin H2, with the help of the enzyme prostacyclin synthase, you can get the prostacyclin. And even from here, with the help of the enzyme thromboxane synthase, you can get the thromboxane. So these are actually the thromboxane, prostaglandin, prostacyclin, or leukotrienes. These are the cell signaling molecules which generally associated with the process of the pain, the process of the fever, the process of the blood clotting, and different types of process in your body. So these are actually the cell signaling molecules which are generally formed from the arachidonate in the human body. Thus you can say that this arachidonate is play a very important part. So I repeat once again. So see, we have started with the acetyl coenzyme A, then converted into malonyl coenzyme A, then upon subsequent addition we have come to get the palmitate. From the palmitate, upon subsequent addition of the carbon, we have finally come to the arachidonate. From this arachidonate you can get different types of cell signaling molecules in the body. So thus you can say this process of the de novo fatty acid synthesis in the human body, it plays a very crucial role so that the body can function in a smooth way. Thus I am going to finish up the part of this de novo synthesis. So thank you everyone for your patience hearing. I hope these slides, this set of these 3 slides, they are helpful for you. If you like my video, you can press this like icon. You can share this video with your classmates and colleagues. Moreover, for further notification from my channel, you can press the bell icon in my channel. So thank you once again to everyone.